joining, but I think it's
So
time.
I have the Cardiovascular Research Discoveries on my screen. Is that comes from Glasgow, right?
Yes,
that's from
this
Glasgow.
is coming from us, and this is during the introduction, and then we will transfer the screen to you. Yeah.
Okay. Very good.
I think we can start if everyone is here. And on behalf of Cardiovascular Research and Council for Basic Cardiovascular Science of the European Society of Cardiology, I would like to welcome everyone very warmly to our second Cardiovascular Research Discoveries seminar. During this seminar, we have an honor to host Professor Klaus Ley and Dr. Clément Cochain, who will talk to us about leukocyte diversity in cardiovascular disease in various aspects. And as you remember, our seminars are organized with very prominent and person who has already contributed hugely to the field, as well as a new investigator that made very recent but very significant contributions as well. And we have an opportunity to listen to their talks, then ask scientific questions. And at the end, as requested by some of our viewers and readers, we give an opportunity to ask questions about study design, about writing perfect papers, as both of our speakers are authors of such perfect papers. And from the point of view of cardiovascular research, we always invite you to submit your best research to our journal, as I think we are really developing into a leading journal in cardiovascular field. So thank you very much. Christian, if I can ask you for a few words and introduction for Professor Ley. Thank you.
Yeah, thanks, Tom. It's a great honor and pleasure to introduce Klaus as an old friend and collaborator. So Klaus is currently professor and director at the Institute, La Jolla Institute of Allergy and Immunology, and also adjunct professor at the University of California, San Diego. Originally, he actually was training at Würzburg University here in Germany, then before moving to the States to become a professor of bioengineering at the University of Virginia and the director of the Cardiovascular Research Center there until 2007, before he moved to San Diego. Klaus has produced and put forward seminal studies on the mechanisms of leukocyte emigration and extravasation in many chronic inflammatory diseases such as atherosclerosis, but also others, as well as important work on myeloid leukocyte homeostasis, and really produced more than 300 original articles and a massive age factor with lots of citations. More importantly, and he's recently basically instigated and spearheaded a joint effort to map the immune cell landscape and atherosclerotic plaques at a single cell level and provide some profiling there. And this is basically how we came up with the program of today's seminar, to have Klaus, give an overview on that, and then also some of his more recent collaborator, Clément Cochain, to follow up on this and then join in a discussion on both talks. So Klaus, the stage is yours. We're very much looking forward to your recent work.
Thank you, Christian, for your kind introduction. Thank you, everybody. Good morning, good afternoon, good evening, wherever you might be around the world. It's a pleasure and honor to give this talk, and I will focus entirely on CD4 T cells. So you should see my PowerPoint screen now.
Yes, we see
ApoB-specific CD4 T cell responses in human cardiovascular disease, and then Clément will talk about the other cells in the order. So, okay. I will talk about protective autoimmunity for a minute, then MHC2 restricted epitopes in human ApoB, testing the ApoB specific CD4 T cell response in cases and controls with tetramers restimulation assays, and now the newest work unpublished, our strategy to find the immunodominant epitopes. So protective autoimmunity, I struggled with this when in 2012, when we discovered that there is a CD4 T cell recall response in the aortic wall, mostly in the adventitia by 2-photon microscopy. And so clearly there's antigen presentation, but that means there is an autoimmune component, but atherosclerosis is not an autoimmune disease. So what is going on? And then later we settled on this and saying atherosclerosis is a chronic inflammatory disease with a secondary autoimmune component. And how do we know this? Well, in all cases, in people with atherosclerosis, we can find antibodies, and we can find CD4 and CD8 T cells that are specific to atherosclerosis antigens. And the same is true in mouse models. So in other words, it doesn't start as an autoimmune disease, but as the disease establishes and becomes chronic, we have always an autoimmune component. That's what I'm studying, because I think that can be manipulated for the benefit of patients. So the first indication was a series of vaccination studies where we basically repeatedly injected ApoB peptides that were MHC class 2 restricted, so you can screen by computer which peptides would be good for CD4 T cells. We did 4 or 5 injections, and then with adjuvant, and then measured atherosclerosis. And what we found in all these studies, that we reduced atherosclerotic lesions by this strategy by 40 to 50%. And we started with 2 peptides called P3 and P6 in 2013. Then we did 3 more peptides. So basically, we looked at different affinities, and it turns out affinity doesn't make a difference. So they all work equally well, 2017. And then P18 in 2018. And this peptide is interesting because it is the only peptide we found that is sequence identical between mouse and human, and also binds mouse and human class 2. So in the Black 6 mouse, the class 2 is IA of B, and in humans, P18 binds very well to DRB10701, which is a fairly common, about 7-8% of population allele. So that makes it tractable. And then we did the controls, no protection by adjuvant only. This is important because there are papers in the field where adjuvant alone has effects. Peptide only with no adjuvant also doesn't work. Irrelevant peptide like MOG or ovalbumin doesn't work. And important for the therapeutic aspect, you can, when we vaccinate the mice after the disease started, it has no effect. It's not bad, it's not good, it just has no effect. So it's not therapeutic, it's preventative. And we'll come back to that when we talk about the vaccine. So the first way to detect these antigen-specific cells was by tetramers. And the tetramer is basically recombinant MHC class 2. Let me change my pointer to laser. Can you see the laser pointer? So it's a recombinant class 2 with a peptide attached to it, and then this is biotinylated, and then streptavidin has 4 binding sites and has a fluorochrome in it, usually APC or PE, and therefore you can find these cells. And this is of the order of 10 to 100 cells per million, so it's not that many. Here's what this looks like. We use 2 colors to improve the specificity of the assay, and here you see a mouse that was vaccinated with P18 and adjuvant, and you see the expansion here from 0.01% to 0.05%, and here you see the statistics for many mice. And what is also interesting is that many of these cells, almost half, express CD25, which is the high affinity IL-2 receptor, and FOXP3, which is the defining transcription factor for regulatory T cells. So back in 2018, we thought, okay, these are probably regulatory T cells. And that makes sense. A regulatory T cell that is specific to an atherosclerosis antigen would be expected to be atheroprotective. Now, how do we find the epitopes in human ApoB? Okay, so this business with the tetramers, it works, but is very cumbersome because there's so many alleles of class 2, and you have to make specific tetramers basically for each class of patient. Even if you take the top 27 alleles that would cover 95% of the population. You would have to make hundreds of tetramers, and it's not easy to validate them, and some fail validation. So in other words, we had to find a more efficient way to do this. So how do we do this? We start with the human ApoB UniProt sequence and then score all the 15 mers against binding for DRB1, 0301, 0701, 1501, and then DRB3 and 4 and 5. Then you get ranks for these peptides. A lower percentile rank means higher affinity. And in the second step, then you produce a median consensus percentile rank using different prediction algorithms. I don't have time to go into the details, but then you get basically the top binders, and we use the top 20%. This results in 195 peptides. So ApoB is very big, it's 4,600 amino acids. So theoretically, you could have 4,580 15-mer peptides, but this narrows it down to 195. And we made all these 195. On top of that, we found a few missense variants. So these are SNPs in ApoB. One of these SNPs, actually is interesting, causes familial hypercholesterolemia. So you probably all know that normally familial hypercholesterolemia is caused by mutation in the LDL receptor, but there is a mutation in the ApoB that can also cause this, because when the ApoB does not bind the LDL receptor properly. And we used mass spec to validate the purity of each peptide. Obviously, what I'm showing you here is a good one, but we had many that were not good, and we had to redo it. Okay, so how do we test this, and what happens in cases and controls? As I said, we had tetramers first, so I will show the old data first. This is now human, DRB1, 0701, ApoB P18. Here is the APC and PE, so the double positive cells are specific. And you can see is in humans, you cannot do a vaccination control, so the best control we can do is a mismatch control, which means we take an individual that does not have this allele. And you see that it's not totally clean. You still have some binding, but there's a significant difference between this and the mismatch. And more than half of the ApoB-specific CD4 T cells in these people express FOXP3 alone. This is flow cytometry data. But in cases with cardiovascular disease, now you get this big orange slice of the pie, and these cells express FOXP3 and ROR gamma T. ROR gamma T is the defining transcription factor of Th17. So this is probably a cell that is underway to move away from Tregs, that at least what we thought in 2018, and becoming an effector cell like a Th17. And you see some also express Tbet, and then they would become Th1. So now this is nice, but it's limited resolution and only 5 transcription factors. That's all you can do with flow cytometry. So now we did a more systematic approach using 10x genomics 5'. We use 5' because that gives us a TCR sequences. You can assemble the VDJ and get the full CDR3. This was led by Ryosuke Saigusa, a postdoc in the lab, and Yanal, bioinformatician, and Kit, the wet lab technician who runs the experiments. Let me show you the validation. So if you plan to work with tetramers, you have to validate. Here is, again, the red and the blue and the double positive cells are here. And the mismatch control, we gated so that the mismatch control was basically contained no cells. And the problems with tetramers are non-specific binding detected by backgating. I don't have time, I only have 25 minutes, so it's in the 2018 paper. But if your tetramer is sticky and binds to monocytes, you cannot use it, right? Because then you don't know whether there's any specific bind. And some tetramers bind to mismatched individuals, so that you can also not use. And if your negative control, which is usually clip peptide that you can get from the NIH, if that binds, well, then you're also in trouble. So in other words, you have to have good validation. And this is a gating scheme. I think I will skip this. I've already talked about the necessity to to have mismatched controls. And here's the backgating. So these are the tetramer positive cells, and they fall when you backgate into the CD3 positive, CD4 positive, as they should, and not into the dump channel. So this is a good one. But I've been in this business now for 5 years, and we don't have more than 2 or 3 good ones, really. Now, as you know, 10x genomics is a hungry beast. It cannot operate on 100 cells. You need basically 1 million cells to start. So therefore, you need carrier cells. And you could use any cell as a carrier cell, but it's better to use a carrier cell that is informative. And we chose to use 3 carrier cells. So here again are the tetramer positive. These are the ApoB specific cells. Then we gated for CD45RA negative. So the naive cells are here, and the antigen experienced cells are here. And of those, we gated for CD25 high, CD127 low or negative. So these are the Tregs. CD25 is the IL-2 receptor, CD127 is the IL-7 receptor. And then of the remaining cells, so that these are not Tregs, you see that big green blob here, you can further differentiate them by CXCR3 because that's a pretty good Th1 marker in human. So here you have your Th1 cells, and here you have your other memory T cells. And then currently, it's not possible to use 10x genomics for rare cells. So there was no method. So we developed a method and patented it. And I won't go into the detail. Our first experiment was actually done by single-cell, single-well, SmartSeq 2, but that's not feasible in the long run because it's too expensive. It's over $50 per cell. So we developed a method with low numbers and with these carrier cells. And we use 4 hashtags. So let me show you the design. So for the tetramer positive cells, you get about 100 cells per patient. For the Th1, about 100,000. For Tregs in our lab, about 50,000. And memory, you can get as many as you want. In this case, 500,000. So then you can do all your washing and your manipulation and your tagging. And then you put them all together and you do the barcoding with cDNA. And then quality controls, and then sequence them on the NovaSeq. A little bit about the clinical side. These samples come from the Women's Interagency HIV study. These women were phenotyped for subclinical atherosclerosis by carotid ultrasound. This is what this looks like. So here is a carotid, and here is a lesion. So we use the lesion count. And we had frozen PBMCs from 8 participants that were DRB 0701 positive. So you remember, I only have a tetramer for 0701, so I had to screen many to find these 8. 2 healthy, 2 with HIV, 2 with no HIV and cardiovascular disease, and 2 with comorbidity. I will not talk about the HIV today, just about the cardiovascular. So we got over 12,000 TCRs for all CD4 T cells, and here you see the clonality. There's very little expansion. So the biggest clone is 61 cells. There's 52, 26, 23, 21. And these are the tetramer positive cells. So we got an output of a total of 110 different clonotypes, and we saw no evidence of clonal expansion. That means they had not recently seen antigen and have been expanded. Only 2 clones were expanded, and not very much, only to a number of 3. And then our Th1 cells, our Tregs and Tmems, also the TCRs are shown here. And the Tmems, the memory T cells, are the most, they have the highest clonality index. And you see the data here. So 44 cells in this talk. The Tregs, by the way, are the most diverse. They have a very low clonality index. So most of these are only seen once or twice. So when you now plot the clonotypes based on the TCR alpha and beta pairs, the tetramers here in black, and you see there are 17 TCRs that are private, that only exist in tetramer positive. But there are almost 80 that are shared with either Th1 or memory, or very few with Tregs, but clearly with memory. So that means that the same TCR can be a tetramer positive, which means an ApoB specific T cell, but it can also be found in the other bulk memory. And we can discuss what that means and why that is. When we plotted our cells in the UMAP, the Tregs were very well defined. So here you have your Treg population, and you can plot this as an isotherm. So you think this mountain here that's the same, different representation of the same data. And that now I project the tetramer positive cells in black on top of the blue mountain. And you see that very few of the tetramer positive cells fall into the Treg region. They're all over the place. When you look at the Th1 and the Tmem, they are not so well separated. So the Th1 are more here on top, and the Tmem kind of a half moon around here. And you see that the tetramer positive cells are all over the place. I don't have time to go into the details, but we have indications that there are other Th1 cells in the Tmems that are not CXCR3 positive. In other words, CXCR3 is not a perfect marker. If you use CXCR3, you will also still have some Th1 cells that you miss, because there are Th1 cells that are not CXCR3 positive. Okay, now about the influence of cardiovascular disease. Here is Th1, Tmem, and Treg, as I've shown you already, but now separated for people with no cardiovascular disease and with cardiovascular disease. And from this, you can calculate the distance of each tetramer positive cell to the center of mass of the Th1, Tmem, and Tregs. And you see here in the CVD negative that the distance is kind of equal. So this is a cumulative histogram. So look here at the 50th percentile. And in the CVD positives, the Tregs, the tetramer positive cells, are further away from the Tregs. To do a statistic significance, we compared tetramer versus memory, and tetramer versus Tregs, and tetramer versus Th1. And you can see there's no difference in the distance to Th1, but in cardiovascular disease, in red or orange here, the cells get closer to Tmem and further away from Treg. So this is significant. This did not quite reach significance. And you can plot this in a ternary plot where you put the three cells here in Tmem, Th1, and Treg, and you can see the people without cardiovascular disease are closer to Treg, further away from Tmem, and in cardiovascular disease, it moves closer to Tmem. So I no longer say that in healthy individuals, the ApoB specific T cells are Tregs. We call them Treg-like because their transcriptome is clearly not identical to Treg transcriptome. Okay, I have about 3 more minutes, and I think I will go straight to the strategy to find the immunodominant. This is published, so I can skip this. The immunodominant epitope. So this is the next project in the lab. Payel Roy is leading this, and we did this first for a small pool of 20 peptides. So we selected peptides that bind a lot of alleles. So here is the number of alleles, DR, DQ, and DP alleles. This is the sum. We require that they bind at least 16. Here is a specificity control. Let's focus on the bottom. CEFX is a collection of epitopes that are viral and from vaccines, and most people have cells to that. So this is a control. When I expand with CEFX and come back and re-stimulate with CEFX, in this LE spot, I get a response, but not with ApoB. When I expand with the ApoB peptide pool and then come back with ApoB, I get the response, but not with CEFX. So this shows specificity. This is a positive control. This is a lectin that you can use to bypass the requirement for specificity. And then when you deconvolve these 20 peptides, you can see actually directly, without even analyzing it, that 2, 11, and 17 look good. And when you do the stimulation index, you see indeed that these 3 peptides are well above the threshold, what we call, there are other peptides that may be immunodominant, but these 3 are clearly causing a big immune response. And when you look at the donors, you see that also many donors, in this case 87%, respond to peptide 2, 11 and 17. So in other words, once you have an immunodominant epitope, then not only you get more cells, but also more people respond to it. And I'm not disclosing the identity of the peptides here because Tomasz told us that this will be on YouTube. So I would like to close by thanking everybody. This project started by Taka Kimura, who is now at Janssen in Tokyo. Dennis Wolf did the more recent work, who's back at the University of Freiburg. Payal Roy and Daniela Weiskopf are leading the effort to find the epitopes. And we couldn't do this without great collaboration with Alex Sette, who runs the IEDB, which is the International Epitope Database. Robert Kaplan gave us the samples from the WISE, and I've mentioned these other people. And I thank you for your attention. Let me stop sharing. And
thank you very much.
Questions.
Thank you very much, Klaus. And we encourage everyone to ask questions through the question and answer section of the of Zoom. And we will have discussion after both of the lectures because in these seminars, our format of discussion is that both speakers are sort of disputing or discussing problems together. And the second speaker in today's seminar is Dr. Clément Cochain from Würzburg, who has worked in Paris Cardiovascular Research Center in France, and his work focused on inflammatory response, neovascularization, tissue repair, and single cell characterization, looking at different cell types. And he will talk to us, I can see, on about the innate immune cell responses, about maybe possibly macrophages and neutrophils that he has contributed through his recent elegant studies. Clément, please.
Yeah, thank you very much for the introduction and for this for the invitation to this very nice seminar. I'm going to change cell type and talk about recent work from my lab, where we focus on the diversity of innate immune cells in the context of myocardial infarction. So it's well known that after myocardial infarction, a strong inflammatory response develops in the heart, involving many cell types. But in this talk, I will focus on neutrophils, monocytes, and macrophages. So this inflammatory response occurs with quite specific kinetics, with in the first phase, disappearance of tissue-resident macrophages, followed by different waves of neutrophils, monocytes, and macrophages. And these different waves of inflammation are linked to various processes of the wound repair process in the heart. So in the beginning, for example, clearance of dead cells, while at later time points, you will have formation of mature scar. And altogether, this inflammatory response in the heart is necessary for this wound healing process and formation of the scar, but at the same time can trigger inflammatory tissue damage and further compromise function of the heart, and also precipitate heart failure in the end. So this dual role of inflammation is directly linked to the functional heterogeneity of innate immune cells themselves in the heart. So monocyte, macrophage, and neutrophils have this wide array of functions, like secretion of different types of cytokines, interaction with other cell types that are involved in many processes, leading either to tissue repair or damage. And you can very roughly group the function of these cells either in kind of tissue repair promoting function and other function that would be more involved in tissue damage. And so given this functional heterogeneity of innate immune cells in the heart, we're indeed very interested in exploring the diversity of these cells to try, for example, to determine whether we can define a subset of functionally distinct neutrophils or monocyte macrophages in the heart. And second, to try and find out what could be the determinants of innate immune cell functional heterogeneity in the heart. So of course, to achieve this kind of aims, the development of single cell technologies in the past years has been really a game changer, and especially for immune profiling, the ability to do so the standard single cell RNA sequencing combined with the analysis of cell surface markers by CytSeq has been really helpful to improve the characterization of immune cells in experimental models, but also in clinical research. So, for example, in a very recent paper, we use single cell analysis to analyze the dynamics of neutrophils in the heart. So here in a mouse model of permanent infarction, we sampled neutrophils at various time points. By single cell, we could nicely see transcriptionally distinct clusters of neutrophils that populated the heart with a quite specific kinetics. So I won't talk too much about that, but the most striking finding that we saw in this analysis was the appearance of neutrophils in the heart expressing this marker CGLEC-F. So here shown at the transcript level, but also validated by flow cytometry. And we saw a time-dependent appearance of these cells, the CGLEC-F plus neutrophils in the heart, and also could see that they had increased effector functions, for example, here, an increased phagocytic activity. So by combining site-seq analysis of neutrophils from the blood and the heart, we could further obtain data showing that the CGLEC-F expression was quite specific to heart neutrophils, and suggesting that this neutrophil state is acquired locally in the heart. So these results were published very recently in this paper, and very recently another very nice paper came to quite similar conclusions. So if you're interested in this story, I would suggest you to check both of these papers. So today I'm going to talk a bit more about macrophages actually, and the diversity in the heart after infarction. So it's quite well established now that in the heart, in the steady state, we can find several types of tissue-resident macrophages that have various developmental origins, and that after an infarct, the landscape of macrophages changes quite drastically, and we can find different heterogeneous type of infarct-associated macrophages. And this change in the macrophage landscape is thought to be mainly driven by the infiltration of monocytes. And so this has been very nicely shown 2 years ago by this paper here, also by single cell analysis of cardiac macrophages here in the steady state heart or 11 days after infarction, where it was nicely demonstrated that the cardioprotective tissue-resident macrophages had quite distinct gene expression signature from the macrophages that we find in the heart here 11 days after infarction. So in my group in recent years and months, we were interested in looking in a bit more detail at the macrophages that populate the heart after infarction, and also at earlier time points after infarction. And recently, so performed site-seq analysis of monocyte and macrophages in the heart here in the steady state and after infarction. And by using the combination of cell surface marker expression and also transcriptomic profiling of the macrophages, we can nicely distinguish subset of tissue-resident macrophages that were described in previous papers, but also some proliferating macrophages, inflammatory Ly6CI monocytes, and different clusters of MI-associated macrophages. And in the remainder of the talk, I will focus more on these, on the main population of infarct-associated macrophages that we found in this data. So by looking at gene expression signature in these different clusters of monocyte and macrophages, we can try to find out a bit more about what could be their function. We can also name them based on enriched genes. So this is sometimes a bit arbitrary, but helps make things a bit clearer for the presentation now. And so besides the inflammatory monocytes in the heart, we found a cluster of macrophages characterized by IL-1 beta expression and MHC2 expression, macrophages with a very clear type 1 interferon response signature, and 2 clusters of macrophages that show enrichment for expression of the receptor TREM2. So if we want to find out what these different clusters or different type of macrophage could be doing there, one thing we can do is look at enriched gene and effectors that have already been validated in the literature. So if you look at these 3 populations, they would fall into more pro-inflammatory pathogenic phenotype. While if you look at the 2 subset of macrophages characterized by TREM2 expression, they express more genes involved in inflammation resolution, erythrocytosis, angiogenesis. They express low inflammatory cytokines, and genes that would be involved in scar formation or fibrosis. So we confirmed that all these MI-associated macrophage population were derived from circulating monocytes. So to this end, we simply depleted circulating monocytes using an anti-CCR2 antibody, and could see that all this population at least of infarct-associated macrophages were clearly reduced when we depleted circulating monocytes. So including these two populations that express TREM2, and I'm going to focus a bit more on in the last minutes of the talk. So what we found interesting about these two populations here is that they displayed a gene expression signature that I would call here a lipid-associated signature that has been already described in other tissue injury contexts and in other disease contexts. That is characterized by the expression of not only of the receptor TREM2, but also other signature transcripts, for example, SPP1, or I just put a set here of the different transcripts that are kind of characteristic of the signature, but there are many others. And this macrophage also express genes in lysosome function, in lipid metabolism, and as I mentioned previously, also express low levels of inflammatory cytokines. So we see now these macrophages with this signature in the heart, but as I mentioned, they were observed already in other settings. So for example, in the setting of atherosclerosis, the so-called TREM2 high or foamy macrophages, and also in other metabolic diseases. So this lipid-associated macrophage signature has been further described in the obese adipose tissue and has been found recently in the fatty liver. And interestingly, you can also see it in the brain in the context of neurodegeneration. The so-called disease-associated microglia also share some of these features. So obviously, the macrophage in the different tissue are not exactly identical, but they share kind of this similar program, which kind of raised the possibility that similar mechanism could underlie the acquisition of this signature. And so recently we tried to dig a bit more into it and try to generate hypotheses as to what could be the drivers of this signature, specifically in the heart. And some possibilities would be, for example, pathways where lipid loading or efferocytosis of dead cells would converge, such as the liver X receptor pathway, which inhibits inflammatory activation of macrophages, and at the same time controls lipid homeostasis, and that we see nicely expressed, at least in a subset of macrophages in the heart. But we focus more recently on the role of the receptor TREM2 itself, since here we can learn from other tissues that acquisition of this specific macrophage signature is dependent on this receptor TREM2. So what we did very recently is that we analyzed the heart of control mice or mice deficient for TREM2 in the steady state of heart infarct, and once again using single cell analysis. And very shortly mentioning the main result that we find in this analysis is that the macrophage subset that show this lipid associated signature, their accumulation is partially blunted in the TREM2 deficient mice, so indicating that accumulation of macrophages with this lipid associated signature is partially dependent on TREM2, as has been shown in other tissues. So yeah, so today, very briefly expose two projects where we kind of use single cell analysis to explore leukocyte diversity in cardiovascular disease, so to find out new aspects of neutrophil dynamics in myocardial infarction, or to identify new regulators of cardiac macrophage states after acute infarction. But of course, there is much more to be learned from this kind of high-dimensional analysis, especially given the constant improvement in technologies that are at our disposition, with also recently the development of spatial transcriptomics, which would be clearly critical to put all this kind of information into the spatial context of the tissue. So yeah, to finish, I would like to thank my team, and especially Giuseppe, who the work that I'm presenting now is his PhD project. And I thank all our collaborators, especially from the Saliba lab and from the Sarnica lab, and all the people that were also involved in this work. Thank you.
Thank you very much, Clément. for a very sort of succinct but very direct and detailed coverage of this wide topic, and it's very impressive. I can encourage everyone to type your questions on question and answer section, and I would ask Christian maybe to start the discussion with our speakers, and panelists can ask the questions directly. Please raise your hand. All the other participants, please type in the question and answer section. Christian.
Yeah, thanks, Tom. So, Klaus, excellent talk. You've already received some praise here from the audience, and there are some questions actually. What actually happens to the Th17 cells? Are they within the TMEM or the Treg compartment in your analysis? And what about other apolipoproteins? Basically, that's the first two questions.
Yeah, I answered a few questions by typing. I don't know whether this goes to everybody or just to the person who asked the question, but let me take these two and answer them in public. So Th17, of course, are of great interest, but the problem is for Th17, there are no surface markers that really work. I mean, you can use CCR6, but CCR6 is also expressed on many Tregs. So it's not a good enough marker. So there's simply no way to sort Th17 cells with current technology. Probably with high dimensional site-seq. So the 10x5' is compatible with that as well. And we have done this with 192 antibodies. So we're currently looking at ways to gate for Th17 cells, but it's not working yet. Now, the reason you can't use RORgamma-T is that once you permeabilize and stain for RORgamma-T, you cannot do single-cell RNA-seq. So you don't get good quality RNA. So that is a limitation of that. And I skipped over this, but I got a question from one of the immunologists. There are really 4 approaches. And Christian, if you allow me to share my screen again, I have a slide that speaks to that. And the slide makes it maybe
I think you can share.
Clear.
You can share, Klaus.
Okay, so let me go up here. I skipped over this because of time, but so there are fundamentally, in addition to the tetramers, there are 4 assays that we use. And I was specifically asked about the AIM assay, the activation-induced marker assay. This is very good because you can use CD40 ligand and CD69, which is our favorite combination. And you get live cells and you can put them in the 10x machine and get the transcriptomes. The downside is here. You need 2 million cells to start. And you know, in some clinical studies, you may not have that many CD4 T cells in a tube. So this is a very low frequency cell. There's no expansion. The transcriptomes are close to what they would be in vivo, and you can, you know, but you don't get enough cells. Now, if you don't have enough cells, you can use cytokine capture assay. So you could say, okay, a Th17 cell makes IL-17A and IL-17F, and you can make an assay with cytokine capture. You also get live cells, and you can also do single-cell RNA-seq on that. And these screening assays I've shown you, ELISpot, we like it because it requires only 100,000 cells. You get massive expansion, but you get no live cells. So it's kind of a dance between how much information you get and how many cells you need to do it. So I think that also speaks to the reason why Th17 is currently not addressable, but we're working on it. Thank you.
I think it also emphasizes that it's important to look using different assays with different ability to detect different functional aspects, because I think transcriptomics, and it's a question probably to both of you that we received by email, is that some of these high throughput techniques are generating a lot of data, but what is the shortest and the best way to functionally understand the functional importance of this remains a question. We identify new populations, but how to identify what they do is a bigger question, I suppose.
If I may, so one way to do this is if you think you have a new cell population, you have to do a lot of validation, because most of these new cell populations are actually doublets. And so you have to be very careful. But if you really have a new cell population, like Clément showed today, let's use the TREM2 high cells, or the type 1, I think the type 1 interferon macrophages, Clément, you discovered those. So let's use those.
No. I just want to, yeah, it's another paper that discovered those in the Heart also from 2017, just to.
Okay. All right. So anyway, so what you need to do, you need surface markers, and surface markers do not correlate with message. In other words, when you plot the messenger RNA for the same gene versus the surface expression, it's not correlated. Very low correlation. There are a few exceptions. CD74 is an exception, correlates well, but most don't. So that means it is important to have the surface markers, as Clément has shown, by CytSeq, and as I said, we use 192 antibodies. And there are technical issues that have to do with the background in the antibodies, that have to do with Fc receptor binding. And we could spend an hour discussing all this, but we don't have the time here today. We are working on a paper right now that compares WNN with gated antibodies. So WNN is a thing from the Satija lab where you can put the CytSeq together, and I know, Clément, you're familiar with that. The CytSeq together with the message. And we're about to compare, well, we have worked on comparing this with another strategy. So in other words, it's not settled. But now, how do you get to the function? Okay, the next step is that you have to find that cell in mice, right? Now, Clément works in mice, so he has a cell right away. In mice, then once you have surface markers, you can sort your cells and can do adoptive transfers, or you can do an in vitro phagocytosis assay or antigen presentation assay or efferocytosis or whatever you want to do, right? So it's kind of a long-winded way of saying to get the function, you have to have an animal model, and not all cell types are the same in humans and mice. And also the markers are not always the same. So if you have a marker that works in human, doesn't mean it's going to work in mice.
I think it ties into a question.
Clément's perspective on that too.
It ties into the question that was asked to Clément, but maybe Clément, do you want to comment on this topic very briefly? And one of the participants asked about the surface markers for your populations.
Yeah, so I agree with Klaus that it's quite complicated then to go for function, but that's also the point of getting this kind of data is, as we are trying to do, maybe find either a surface marker that you could target, or pathways that you could target to make this population or this one disappear or expand, and then try to look for what are the functional outcomes afterwards. And regarding surface markers for these macrophages, unfortunately, there are no clear surface markers. So that's a bit difficult with macrophages in the heart. There are very good markers for some subset of resistant macrophages, which we find also by CytSeq. You have tons of markers that you can use and look at this population. What I can say is that these population of monocyte-derived macrophages have some function, at least from the data that we have. You could gate them by, let's say, by excluding the other population. But then again, you will not go and be able to discriminate the subcluster that I've shown here, which is why we have sometimes to go back to doing single cell in your wild type and your knockout and in your different conditions to be able to really nicely define these clusters. And of course, I call these cells, they express TREM2 at the RNA level, but that's one of the difficulties that it's not really detectable at the surface because it's sensitive to enzymatic digestion, and the antibodies that are available are bad. So yeah, this is not
it's challenging. Before I pass to Federica Marelli-Berg, who has a question, and Christian to continue the discussion, there is also another question to Clément about the modulation of these populations pharmacologically. And the person asking is asking about lipid-lowering drugs, but I would widen it to potential pharmacological agents that could target TREM2.
Yes, so there are stuff that are in development, and some of them have been published. I think that that would activate specifically this pathway, the TREM2 pathway. So there is a nice review from the lab of from the AMIT lab on this topic in Cell, like from last year, I think. And the question about lipid-lowering drugs, I have no idea, but that's a nice hypothesis, of course, to check what would be the effect of lipid-lowering drugs on macrophage populations in the heart. Yeah, now that we have these technologies to have more precise notions about what could be going on. Yes.
Thank you.
We don't know about macrophage populations in the heart yet, but what we do know is that statins, lipid-lowering drugs have huge effects on monocyte gene expression. And this is actually published. This is Ehinger 2020 from my lab. And we did 92 patients. And it was a chance discovery. I didn't expect this, but there was a confounder, and you know, we couldn't figure out what the confounder was. And it turned out when we analyzed it, the confounder was statin use. And statin use has a huge effect on the transcriptome in monocytes. In this paper, it's classical monocytes, but there's also signatures in intermediates and non-classical monocytes. So in other words, the statin effect, the so-called pleiotropic statin effect, or the overall statin effect, is very visible in the transcriptome of monocytes. That's all I can say for sure right now.
On one hand, gives therapeutic value. On the other, shows how important it is to carefully design your clinical populations and studies, because if you may easily overlook statins if you are an immunologist, and one shouldn't. So maybe we can ask Federica to ask the question, and then Christian to continue the discussion.
Yes, thank you to both speakers. Fantastic presentation. I just have a question for Dr. Ley. That is, these tetramer positive cells, I will be specific. Do you know how this particular immune response developed with the progression of atherosclerosis? And can you work out a causative role of these cells? For example, I was thinking there are models of atherosclerosis regression in mice. Was wondering whether you thought about that. Thank you.
Thank you, Federica. That's a great question. So we know a little bit about this. Some of it is published. So let me, in mice, we know that the population of ApoB specific CD4 T cells expands early in an ApoE mouse. When you put them on Western diet, within a week or two, you get a large expansion. And then slowly these cells, the cell population collapses. So in a year-old mouse, you find very few. Now, maybe we didn't look in the right place, because T cells have many functions and go in many places. So some epitope-specific T cells are more found in lymph nodes. Others are more found in the spleen. Yet others are follicular helper T cells, and they're found in germinal centers where they support isotype switching of the B cells. Another way you can manipulate the system is this vaccination approach. And we've done this now for 7 years, and we had trouble finding the cells that expanded after vaccination. So it was not lymph node, it was not spleen, didn't see anything in the bone marrow. And then after, basically looking through the whole mouse, we found them in the peritoneal cavity. That's not where you normally expect CD4 T cells, but that's where they expand. I showed you 2 data slides on this. So they expand, and many of them become Tregs initially. So the immune response to self-antigens is not well understood outside of autoimmune diseases. Even autoimmune disease is not well understood. You're supposed to not have these T cells because of negative selection, but then, as Mark Davis taught us, negative selection is not completely effective. So you have these cells, and we can find these cells, but the response to the self-antigens might be quite different from the response to foreign antigens. So we have a little bit of time course, and what we saw there were 2 factors, age and diet. So we could see a signature that was induced by diet, high-fat diet or Western diet in the case of ApoE. and another thing that was age, and they were almost orthogonal. So when you plotted them by principal component analysis, they were not the same genes, they were different genes that were affected. Thank you.
Klaus, maybe following up on that. I mean, you've seen the high throughput B cell cloning for autoantibodies resulting in this mitochondrial dehydrogenase as an autoantigen. So I'm actually wondering, given all the complications that the tetramer issue confers here, whether you would also consider a more unbiased profiling of TCRs, you know, in your model to maybe come up with some completely unrelated autoantigen that might work as well. Yeah?
Yeah, that's a great question, Christian. So we actually doing this. We're using the Saber Cell. The Saber SABR was developed in David Baltimore's lab by Alok Yogelkar, and he's now a professor in Pittsburgh, and so we work with him. So the Saber Cell, in a nutshell, is a cell that where the MHC class 2 or class 1 is hooked up to an NFAT reporter, NFAT GFP, and then you basically transfect in a library with your peptides, and then you interrogate human or mouse T cells, and you find for which T cells you actually have, for which antigens you actually have T cells. And because you can then sequence your library, you know what the antigen was. This is a great approach, unbiased. We've worked on it for about 2 years, and there's still we still have signal to noise issues. There's an alternative approach, I think, from Munich where they pick single cells. So there are about 3 or 4 such approaches, and yes, this is where we need to go, but I have not seen any convincing results in atherosclerosis for T cells yet.
So maybe one more. Thanks, Klaus. This was a great answer. So maybe one more question to Clément. This has also come up here in the Q&A. I mean, you have identified a strand 2 high population, and probably the best characterized effects are in Alzheimer's disease, where basically TREM2 loss of function risk variant actually exacerbating disease and causing, in particular under ApoE deficient conditions, like microglial changes that cause cholesterol ester accumulation, for instance. So I actually was wondering, and the question also relates to that, what happens in the TREM2 knockout in myocardial function functionally and concerning your phenotype, if you
so this is a
regulation of lipids.
Yeah, this is of course a very important question, and this is currently under investigation. Some showing you very recent data, and so from their gene expression profile and other disease contexts, we would expect a protective role of the cells, but I don't have a clear answer to give you to that, and
So these are ongoing then.
Yeah.
Okay.
One more question that we received by email that is more sort of related to philosophy of designing a study is that Klaus was talking about more human data and Clément mouse data. How do you balance the clinical approach to then bring down the mechanistic approach in the animals? What is the perfect balance? Of course, it's a difficult question to answer, but is it better to start with a human characterization, which is by default difficult because of the clinical features that, as Klaus indicated, might include variables that we have no control over, or is it better to start with mouse? That's what one of the participants was wondering. I don't know who wants to say first. Klaus?
Clément, I mean, you want to take it?
Yeah, you can.
Okay, I can. We do both. So my lab was a mouse lab for 20 years or so, and we started the human work about 10 years ago. And as I said in an earlier comment, you really need both, because only in the mouse model can you do the mechanistic work. And it turns out that translating the markers both the gene signatures and, even worse, the surface markers from mouse to human or human to mouse is not trivial. It's much more difficult than I had anticipated. So you're often struggling, with even mapping a cell type from one species to the other. But you have to do it because to find out what your cell does, you have to have all these assays, adoptive transfers, or you put in a DTR and then you deplete the cell and see what happens in your disease model. So the second thing I would say is it is not necessary that the same person does both, but it is very good to talk to each other. So if someone is purely a clinical investigator and only looks at PBMCs from their cohorts, right? These are great collaborations with working with someone like Clément who has all the mouse models. And now it is even more complicated because the single cell RNA-seq has taken on a life of its own, and maybe Clément should comment on this. This is so complicated that the team to do the work that I showed today. So basically, I always put together a biology postdoc, a wet lab technician who runs the experiment, a bioinformatician, and then someone to integrate it. Often a clinical statistician or epidemiologist. If the cohort is complicated, we need that too. So it's like 5 people to get this work done, right? So it's pretty complicated.
Clément, do you want to comment?
Yeah, I fully agree with Klaus that both are necessary when it comes to what I talked about mostly today. So single cell analysis and so on. I think that it's also now a good opportunity to have data where we can, in a more precise way, try to compare what's going on in the human situation, in the mouse situation. So we try to do that. For example, recently in the context of atherosclerosis, try to compare what kind of disease-associated signature we can see in macrophages in mouse and humans, and try to find out what are the, let's say shortly, the commonalities and differences. And I think it's been done in a tons of diseases, and data being generated from clinical studies, single cell data, by big initiatives like the Human Cell Atlas and so on, that anybody can access and kind of look at this data and try to find out whether the pathway that they are looking at in mice might be also relevant in humans. So I think that these kind of new technologies that we get now are a really good opportunity to bridge the mouse models and the clinical situation also.
And partly related to this, and maybe it's actually also a question to both of you, is that if you do such characterization in human, sort of an inbuilt problem is a small number. Yeah, Klaus, you showed a comparison of 2 examples of patients, one with CVD, one without CVD. But in human samples, you need n numbers. So how do you marry these 2 things? How do you get sufficient n numbers without going bankrupt and providing a strong enough data in a clinical setting? How do you validate? Maybe a question taken directly from one of the participants.
Yeah, this is a very important question. So single cell RNA-seq is a discovery tool, and is not suitable for large cohorts. And we get dinged even in one study we had 92 patients with. So bulk sequencing of sorted cells is better than single cell RNA-seq because it gives much deeper transcriptomes. People often don't appreciate that. They think, okay, bulk RNA-seq is bad because it's older, but it's not. It's actually better. Once you discover what you needed to discover by single cell and site-seq, then you can, once you find a way to sort your cell of interest, you should do a bulk RNA-seq. And bulk RNA-seq is cheaper and better. You get 20,000 genes and you have a much better transcriptome than any single cell technology. So it's kind of iterative, and I would say, so I work with 2 groups of clinicians, one in the WISE study, another one in the CAVA study, that in general the clinical investigators are not yet attuned to these technologies, and they mostly use flow cytometry, even with low number of lasers and colors. So it's a challenge, and it will not happen overnight.
Yeah. Clément?
I have no further comments on the.
Okay, I think it was an extensive and good answer. Christian, are there any other questions? Any panelists or anyone else? We will make sure that all the questions that you've asked on the question and answer section of this Zoom that were not asked directly to our speakers will be transferred to them, and we will sort of be an intermediary to send you the answers. So we will provide the answers to all the questions which we couldn't ask for the sake of time. Christian?
I think we've covered most of the scope of the questions, and I think, as you said, you know, the rest should be settled in a more detailed or direct context.
So I think on behalf of both of us, I would like to thank our speakers for excellent presentations, but also really great discussion on that fits perfectly to the title of the series, Cardiovascular Research Discoveries, because it's exactly focused on how to discover new players in immune mechanisms of cardiovascular disease. So it was very exciting to all of us, I think. Thank you very much for joining, and thank you to all the attendees who joined us today. And we had over 350 attendees during the talk, so it's very good. Thank you for joining, and please join us for the next seminar that we will communicate to you soon. Thank you.
Looking forward. Cheers.
Thanks.